3.3 Intercellular junctions allow adjacent cells to adhere or communicate. Learning objective 3.3.1 compare the structure and function of tight junctions, desmosomes, and gap junctions. In many cases, the plasma membrane of adjacent cells are joined together by specialized cell junctions that allow neighboring cells to adhere and sometimes communicate. These junctions may aid or inhibit movement of molecules between or past cells and also serve to tie cells together into tightly knit communities. Let's look at each of the three types of cell junctions. So this is figure 3.4 titled Cell Junctions. We'll start with tight junctions. Tight junctions are impermeable junctions. They form continuous seals around the cell. They prevent molecules from passing between cells, and they create two compartments, one on each side or each layer of cells. Desmosomes are anchoring junctions. They bind adjacent cells together like molecular Velcro. You can think of them as like a little square of Velcro. And they help keep cells from tearing apart. Last is gap junctions. These are communicating junctions that allow ions and small molecules to pass from cell to cell. These are particularly important in heart and embryonic cells. Tight junctions. In a tight junction, a series of integral protein molecules in the plasma membranes of adjacent cells are fused together like the zipper of a Ziploc bag. This forms an impermeable junction that encircles the cell, so it goes all the way around. It creates two fluid-filled compartments separated from each other by an impermeable layer of cells. Tight junctions help prevent molecules from passing through the extracellular space between adjacent cells and restricts movements of membrane proteins. For example, tight junctions between epithelial cells lining the digestive tract keep digestive enzymes and microorganisms in the intestine from seeping into the bloodstream. Although called impermeable junctions, some tight junctions are leaky and may allow certain ions to pass. Desmosomes. Desmosomes serve as anchoring junctions, mechanical couplings scattered like rivets along the sides of adjacent cells to prevent their separation. On the cytoplasmic face of each plasma membrane is a button-like thickening called a plaque. Adjacent cells are held together by thin linker proteins called cadherins that extend from the plaques and fit together like Velcro in the intercellular space. Thicker keratin filaments, known as intermediate filaments, which form part of the cytoskeleton, extend from the cytoplasmic side of the plaque across the width of the cell to anchor to the plaque on the cell's opposite side. In this way, desmosomes bind neighboring cells together into sheets. Desmosomes also anchor a continuous internal network of strong fibers that distribute tension throughout this cellular sheet and reduce the chance of the sheet tearing when it is subjected to pulling forces. Desmosomes are abundant in tissues subjected to great mechanical stress, such as skin and heart muscle. Gap junctions. A gap junction is a communicating junction between adjacent cells. At gap junctions, the adjacent plasma membranes are very close, and cells are connected by hollow cylinders known as connexons, composed of transmembrane proteins. Different types of gap junctions are composed of different transmembrane proteins. They determine what can pass through them from one cell to its neighbor. Ions, simple sugars, and other small molecules pass through these water-filled channels. Gap junctions are present in some electrically excitable tissues such as the heart and smooth muscle, where ion passage from cell to cell helps synchronize their electrical activity and contraction. Substances move through the plasma membrane in essentially two ways: passively or actively. In passive transport, substances cross the membrane without any energy input from the cell. In active transport, the cell provides the metabolic energy, usually ATP, needed to move substances across the membrane. Active and passive transport processes are the topics of the next two modules, summarized in tables 3.1, 3.2, and 3.3. And that is 3.3.